What if the most sophisticated quantum computer exists inside your cells? Scientists have discovered that DNA may operate using quantum mechanics, the strange physics where particles exist in multiple states simultaneously, essentially making your cells function like miniature supercomputers hiding in plain sight. In 1944, physicist Erwin Schrödinger asked in his book, "What is life?" How could the laws of physics explain living organisms? Today, quantum biology suggests an answer that challenges our fundamental understanding of evolution. Could the mathematical complexity of our genetic code point to something beyond mere chance? The answer might redefine our very understanding of life itself. Schrödinger's revolutionary question. In 1944, while World War II raged across Europe, a renowned physicist was asking a question that would eventually transform our understanding of life itself. Erwin Schrödinger, who had already revolutionized physics with his wave equation, turned his attention to biology. His lectures at Trinity College Dublin, later published as the book What is Life?, posed a question that seemed simple but carried profound implications. How could the physical laws that govern atoms explain the behavior of living organisms? Schrödinger recognized a fundamental problem. Classical physics, the physics of Newton that explained the motion of planets and objects we can see, failed to explain how genetic information could remain stable across generations. At a time when genes were still a somewhat abstract concept and the structure of DNA remained unknown, Schrödinger made a remarkable intuitive leap. Approaching biology through the lens of quantum mechanics, the strange new physics he had helped create, Schrödinger proposed that genetic material must be structured as an aperiodic crystal, a stable molecular structure containing vast information in its irregular arrangement. This prediction came 9 years before Watson and Crick discovered DNA's double helix structure. Like a bridge spanning two distant shores, Schrödinger connected the mathematical world of physics with the organic complexity of biology, creating a pathway future scientists would follow. In the 1940s, biology and physics existed as separate disciplines with little crossover. Biologists studied whole organisms using microscopes and dissection, while physicists worked with equations and particle accelerators. The application of quantum physics to living cells seemed far-fetched to scientists in both fields. Schrödinger was creating a new scientific territory. He wrote that understanding life would require new physics, not because existing physics was wrong, but because biology presented challenges that would expand our understanding of physical laws. This interdisciplinary approach predated by decades the merging of fields we now take for granted. The reception to his ideas mixed skepticism with fascination. Many biologists dismissed his theories as speculative overreach from a physicist unfamiliar with living systems. Some physicists worried that applying quantum mechanics to biology would undermine the mathematical rigor of their field. Yet others were intrigued by the questions he raised. What made Schrödinger's work so prescient was his specific predictions about the molecular basis of heredity. He correctly reasoned that genetic information must be encoded in a molecule stable enough to preserve information over generations, yet variable enough to allow for the diversity of life. He estimated how much information such a molecule would need to contain and speculated about its structural properties. When Watson and Crick discovered DNA's structure in 1953, the molecule matched Schrödinger's conceptual description with remarkable accuracy. DNA is indeed an aperiodic crystal with a variable sequence of bases encoding genetic information. The double helix contains billions of chemical letters forming the genetic instructions for building and operating living organisms. Schrödinger had been right about information storage too. He recognized that life requires a physical system capable of containing enormous amounts of information in a stable form. DNA's four-letter alphabet provides this capability, with a single human cell containing about 700 megabytes of genetic information. Despite these insights, technological limitations prevented scientists from testing whether quantum effects actually operate in biological structures. The instruments needed to detect quantum phenomena in complex molecules simply didn't exist. Experimental verification of his hypothesis would wait decades. Through the following decades, molecular biology advanced rapidly with discoveries about DNA transcription, RNA, and protein synthesis. This central dogma seemed to explain life's fundamental processes without needing to invoke quantum mechanics beyond its basic role in chemical bonds. Schrödinger's more radical suggestions about quantum effects in biology faded into the background. The technological barriers were significant. Quantum effects occur at extremely small scales and last for incredibly brief periods, often less than a billionth of a second. Detection requires specialized equipment that wasn't developed until the late 20th and early 21st centuries. Schrödinger made these predictions without modern computational tools. Today, scientists use supercomputers to model quantum interactions in biological systems. He worked with pen, paper, and mathematical intuition developed through his fundamental physics work. His insights extended beyond genetic material structure. Schrödinger questioned how enzymes could catalyze chemical reactions with such precision and efficiency, suggesting quantum mechanics might explain these biological catalysts, another prediction modern research is now confirming. It took nearly 60 years for technology to advance enough to begin testing Schrödinger's hypotheses about quantum biology. With ultra-fast lasers, sensitive spectroscopy, and quantum sensors, researchers began detecting phenomena like quantum coherence, tunneling, and entanglement in living organisms. Evidence now confirms Schrödinger's suspicion: DNA functions as a quantum system in specific ways. The electron arrangement in DNA bases creates conditions where quantum effects influence mutations, DNA repair, and possibly information storage. The hydrogen bonds holding DNA's double helix together exhibit quantum properties affecting the genetic code stability. Schrödinger's question, what is life? Opened a door scientists continue to explore. His suggestion that quantum mechanics plays a fundamental role in biology launched what became quantum biology, examining how quantum effects influence processes from photosynthesis to bird navigation, enzyme function, and possibly consciousness itself. These discoveries are transforming our understanding of biology at its most fundamental level. The quantum nature of life that Schrödinger proposed is moving from theoretical speculation to experimental reality, opening new frontiers in both biology and physics. This convergence raises profound questions about life's nature and origin, revealing that plants and other organisms have been harnessing quantum effects all along. The quantum nature of photosynthesis. Plants across our planet perform a remarkable feat every day that may rely on quantum mechanics. Photosynthesis, the process that converts sunlight into usable energy, operates with an efficiency that has puzzled scientists for decades. The question emerges, have plants been using quantum computing principles to harvest sunlight for billions of years? Photosynthesis stands as perhaps the most fundamental biological process on Earth. Without it, the oxygen-rich atmosphere that supports animal life wouldn't exist. Plants convert incoming sunlight to chemical energy with efficiency rates approaching 95% in the initial energy transfer steps. This level of efficiency exceeds what human-engineered solar panels can achieve, which typically convert only 15 to 20% of sunlight into electricity. For most of scientific history, researchers understood photosynthesis through classical physics models. The dominant theory, known as force-through-resonance energy transfer, FRET, explained how energy moves from molecule to molecule when a chlorophyll molecule absorbs a photon of light. While this model explained basic observations, it couldn't account for the extraordinary efficiency rates. Classical physics predicted far more energy loss as heat during these transfers than what scientists observed. This efficiency puzzle led researchers to suspect something beyond classical physics must be happening. Plants somehow avoid the random, wasteful pathways that energy would typically take. It was as if the energy identified the most efficient path rather than bouncing around randomly. In 2007, a groundbreaking nature study provided evidence challenging the classical understanding. Scientists studying the phenomenon of Diffusion FMO, complex in green sulfur bacteria, discovered something extraordinary. This protein complex serves as a molecular wire conducting energy from light-gathering antennas to the reaction center where chemical conversion occurs. The FMO complex consists of 3 identical protein subunits, each containing 8 precisely arranged bacteria chlorophyll molecules. These molecules form a molecular circuit board with their specific positioning critical for the quantum effects that researchers discovered. The study revealed that excited energy states in the FMO complex didn't simply hop sequentially from molecule to molecule. Instead, the energy existed in multiple locations simultaneously, a phenomenon called quantum coherence. This mirrors quantum behavior that enables quantum computers, where quantum bits exist in multiple states simultaneously rather than just binary 0 or 1. Using femtosecond multidimensional spectroscopy, employing incredibly short laser pulses, researchers tracked nearly instantaneous energy movements. These experiments revealed quantum beating, oscillations indicating quantum coherence. Remarkably, this coherence persisted for up to 660 femtoseconds at room temperature, far longer than theories predicted possible in biological environments. The energy in photosynthesis behaves like a driver who can test all possible routes on a highway simultaneously before instantly materializing on the fastest path. Rather than trying each path sequentially like classical navigation, the energy wave explores all paths in parallel before selecting the optimal route, essentially performing a quantum computation to maximize efficiency. What makes this discovery astonishing is that quantum coherence was thought to require highly controlled environments, near absolute zero temperatures in a vacuum. Biological systems with their warm, wet, constantly moving molecules seemed incompatible with quantum effects. Yet, evidence shows the protein structure might actually protect and enhance these quantum effects rather than destroying them. The protein scaffold vibrations appear tuned to the right frequencies to support quantum coherence. Imagine an orchestra where molecular vibrations play in perfect harmony, creating a protective environment for quantum processes rather than disrupting them. Evolution seems to have optimized these proteins for both their chemical and quantum mechanical properties. Similar quantum phenomena have been observed in various photosynthetic complexes across different species, from ancient cyanobacteria to modern plants. This suggests quantum mechanics is a widespread strategy in nature for efficient energy capture and transfer. These discoveries have sparked new research directions in both quantum physics and biology. Engineers now study these natural quantum systems to design better solar cells and energy technologies. If we can replicate nature's quantum methods, we might develop artificial photosynthesis systems with similarly high efficiency. The discovery of quantum effects in photosynthesis challenges our understanding of evolution. Has natural selection been optimizing quantum processes for billions of years, long before humans understood quantum mechanics existed? This finding breaks down the conceptual barrier between quantum and biological worlds. The warm, wet environment of living cells no longer seems incompatible with quantum effects. This insight has opened investigations into other potential quantum phenomena in biology, from enzyme catalysis to DNA mutation mechanisms. The quantum nature of photosynthesis suggests life may be intrinsically quantum mechanical at its core. Plants haven't been using an analog of quantum computing, they've been performing actual quantum computations in their cells for billions of years. As we continue exploring quantum foundations of biology, another surprising area has emerged. The role quantum mechanics plays in how enzymes work, particularly in DNA itself. Quantum tunneling in enzymes. Inside your body right now, billions of chemical reactions are occurring that, according to classical physics, should be impossible. These reactions happen not because the laws of physics are being broken, but because they're being bypassed through a quantum mechanical phenomenon called tunneling. This process is particularly crucial in enzymes, the biological catalysts that make life possible. Enzymes are specialized proteins that accelerate chemical reactions in living organisms. Without these molecular machines, the reactions necessary for life would proceed too slowly to sustain biological functions. Your body contains thousands of different enzymes, each designed to facilitate specific chemical transformations. They're responsible for everything from breaking down the food you eat to replicating your DNA when cells divide. Enzymes increase reaction rates by factors of millions or even billions compared to uncatalyzed reactions. This acceleration allows processes that would naturally take years to occur in fractions of a second. Understanding how enzymes achieve this remarkable efficiency has puzzled scientists for decades. Chemical reactions require molecules to overcome energy barrier, like climbing a mountain before transforming into products. This barrier, the activation energy, determines reaction speed. Traditional enzyme theory explains that enzymes lower this barrier by holding reactants in optimal positions. However, this explanation only partially accounts for enzyme efficiency. Scientists consistently observed reactions occurring hundreds of times faster than classical models predicted, particularly for hydrogen atom transfers common in biological systems. The solution came quantum mechanics and the phenomenon of tunneling. In the quantum world, particles exist as probability waves rather than definite objects. This wave-like nature allows particles to occasionally pass through energy barriers instead of going over them. Imagine a shortcut through the mountain rather than climbing over it. Quantum tunneling creates these impossible pathways. Hydrogen atoms, being the lightest elements, exhibit the strongest tunneling effects. Their small mass gives them a more pronounced wave-like character, increasing their tunneling ability. This property becomes critical in many enzyme reactions involving hydrogen transfers. In 1989, scientists at MIT conducted a groundbreaking experiment with the enzyme alcohol dehydrogenase, which breaks down alcohol in your liver. They replaced normal hydrogen atoms with deuterium, a heavier hydrogen isotope. If classical physics alone governed the reaction, this substitution would cause only minor slowdowns. Instead, they observed reaction rates dropping dramatically by factors of 50 or more. This provided compelling evidence that quantum tunneling was essential to the enzyme's function, as the heavier deuterium atoms couldn't tunnel as effectively as hydrogen. DNA polymerase demonstrates this quantum advantage remarkably. This enzyme replicates DNA with an error rate of less than 1 in a billion. Like a quantum courier taking shortcuts through an impossibly congested city, hydrogen atoms tunnel between DNA bases, allowing the enzyme to check and correct potential errors with extraordinary speed and precision. Without tunneling, DNA replication would be too slow and error-prone to maintain genetic stability. Scientists have used advanced spectroscopic techniques to directly observe hydrogen tunneling in enzymatic reactions. These methods track atom positions with extraordinary precision, showing that hydrogen atoms don't follow classical paths, but instead tunnel through energy barriers. Enzymes haven't just passively adapted to quantum effects, they've evolved to optimize them. Their structures create environments that position donor and acceptor atoms at ideal distances for tunneling. Protein dynamics, subtle movements within enzyme structures, bring reactants into alignments that maximize tunneling probability. This fine-tuning suggests that quantum effects aren't incidental, but essential to enzyme function. Beyond DNA polymerase, quantum tunneling appears in enzymes involved in cellular respiration, photosynthesis, and various metabolic pathways. Soybean lipoxygenase shows some of the most dramatic tunneling effects ever measured in a biological system, accelerating hydrogen transfer rates by over 1,000-fold compared to classical predictions. Quantum tunneling contributes to both DNA mutation and repair. Occasionally, hydrogen atoms and DNA spontaneously shift positions through quantum effects, creating tautomeric forms, alternative arrangements of the same atoms that can lead to mismatches during replication. These quantum events contribute to the background mutation rate driving evolution. Repair enzymes then use tunneling themselves to identify and fix these changes, creating a balance between genetic stability and evolutionary variation. This quantum view transforms our understanding of life at a fundamental level. Biological systems have evolved to harness and control quantum effects. The precision of DNA replication, the efficiency of metabolic processes, and ultimately the stability of the genetic code all depend on quantum tunneling. The discovery that life depends on quantum effects raises profound questions about how such systems evolved. The enzyme structures facilitating quantum tunneling require precise atomic arrangements encoded in DNA, which itself relies on quantum tunneling. This creates a remarkable interdependence where quantum effects enable the very systems that have evolved to harness them. When considering life's origin, we must account for the emergence of structures optimized for quantum mechanical processes. This adds significant complexity to mathematical models calculating the probability of life arising through random processes. As we'll see next, when these quantum processes are incorporated into calculations about DNA's formation, the mathematical picture becomes considerably more intricate. The mathematical improbability debate. The mathematical challenge of calculating the probability of life's emergence has fascinated scientists and philosophers for generations. Now, with our growing understanding of quantum biology, this calculation has become significantly more complex. When we try to determine the odds of quantum-capable DNA forming by chance, we enter mathematical territory that stretches the limits of probability theory. Traditional biological structures are already remarkably intricate. A single human cell contains approximately 30,000 different proteins, each folded into precise three-dimensional shapes necessary for their function. DNA itself consists of billions of nucleotide bases arranged in a specific sequence. The probability of such complex structures forming randomly has long been debated in evolutionary biology. Quantum biology adds additional layers of complexity to these calculations. The physical arrangement of atoms and molecules matters critically alongside their quantum mechanical properties. For DNA to function as a quantum system, it requires precise arrangements of electron clouds, specific distances between atoms that enable tunneling, and molecular structures that can maintain quantum coherence. These quantum requirements significantly constrain the possible arrangements that would result in functional biological systems. Consider finding a specific grain of sand on all the beaches of the world. This gives us a glimpse of the probabilities involved in forming complex biological structures randomly. Several mathematical models have been used to calculate these odds. The simplest approach multiplies the individual probabilities of each component forming correctly. For a moderate-sized protein of 300 amino acids, with 20 possible amino acids at each position, this calculation yields a probability of 1 in 20 to the power of 300, an astronomically small number. When quantum requirements enter the equation, the probabilities shrink further. Quantum tunneling in enzymes requires atoms positioned at precise distances and angles. Maintaining quantum coherence demands molecular structures that shield quantum states from environmental decoherence. These constraints dramatically reduce the mathematical probability of such systems forming by chance alone. This mathematical territory has become central to debates about life's origins. Proponents of intelligent design highlight these calculations as evidence for their perspective. They point to probabilities like 1 in 10 to the power of 183 for specific protein sequences forming randomly, concluding that such improbability points to deliberate design rather than chance. However, evolutionary biologists offer substantial counterarguments that transform the probability landscape. Natural selection fundamentally changes these calculations. While mutations occur randomly, selection pressures consistently favor certain outcomes, making evolution a non-random process. Instead of requiring all components to form simultaneously by chance, evolution works through incremental steps, each providing some survival advantage. The vast time frames available for evolutionary processes also alter probability assessments. Life's 3.8 billion year history on Earth provides enormous time for evolutionary exploration. With trillions of organisms existing simultaneously, each potentially experiencing mutations, the probability space has been extensively sampled. What seems improbable in human timeframes becomes more reasonable over geological time. Selection pressure makes improbable structures achievable through natural processes. Even small improvements in function can provide survival advantages. Once a basic function emerges, selection can rapidly refine it. This directed optimization means evolution doesn't blindly search the entire possibility space. It follows paths of increasing fitness, significantly improving the odds of complex structures forming. Computer simulations confirm this, demonstrating that when selection is applied after each random step, complex structures reliably develop from simple starting conditions. These models suggest quantum-capable biological systems could evolve through similar processes of incremental improvement under selection pressure. A critical error in probability calculations involves what researchers call the post hoc probability fallacy. This occurs when calculating the probability of a specific outcome after it has already been observed, like calculating the odds of a specific protein sequence we see today. This approach ignores that many different sequences could fulfill the same function, and evolution had no predetermined target. As research notes, in most cases, the post hoc fallacy completely nullifies the argument. The relevant question isn't what are the odds of this exact arrangement forming, but what are the odds of some functional arrangement forming? Genetic redundancy and duplication further enhance evolutionary probability. Many biological structures evolved from simpler precursors through gene duplication followed by specialization. This pathway allows complex systems to develop without requiring simultaneous random formation of all components. Quantum biology introduces additional questions about when quantum effects became integrated into biological processes. Did quantum tunneling in enzymes emerge early in evolution? Was photosynthetic quantum coherence a gradual development? These questions affect how we model the probability of quantum biological systems emerging. Modern mathematical models must account for both classical and quantum mechanisms when calculating evolutionary probabilities. Classical mechanisms like natural selection interact with quantum processes like tunneling and coherence, creating a more nuanced picture than either pure chance or predetermined design suggests. The mathematics of probability in evolution resists simple either-or propositions. Complex biological systems with quantum properties likely emerge through a combination of random variation, non-random selection, and potentially other factors not yet fully understood. The improbability of such systems forming in a single random step doesn't preclude their formation through evolutionary processes over billions of years. As our understanding of quantum biology expands, our mathematical models of how these systems could have evolved will become more sophisticated. The direct evidence of quantum behavior in DNA provides crucial insights into how genetic information is stored,
quantum
processed,
information
and protected at the most
When we
fundamental
look at a
level of
strand
biology.
of DNA through the lens of quantum mechanics,
An entirely different picture emerges than what we see in biology textbooks. The familiar double helix structure, with its ladder of base pairs connecting the two strands, becomes a sophisticated quantum system with properties that might enable computational abilities far beyond what classical biochemistry suggests. DNA functions like a biological quantum computer, with each nucleotides serving as a potential quantum bit rather than a simple chemical building block. The 4 nucleotide bases, adenine, thymine, guanine, and cytosine, pair specifically to form the rungs of the DNA ladder. At the quantum level, each base contains rings of atoms with delocalized electrons forming electron clouds. These clouds create quantum states that can exist in superpositions, occupying multiple states simultaneously. The quantum states arise from the arrangement of electrons in the molecular orbitals of DNA bases. The pi electrons form clouds, above and below the plane of the base rings, and can occupy multiple energy states as probability distributions, creating the foundation for quantum behavior within DNA. Research shows that up to 1 in 100 mutations in DNA may be directly attributed to quantum tunneling effects, suggesting quantum mechanics plays a significant role in genetic evolution. Quantum tunneling appears during DNA mutation and repair processes. While classical physics requires particles to have sufficient energy to overcome barriers between states, quantum particles can tunnel through energy barriers. This occurs in DNA when protons and hydrogen bonds between base pairs shift positions. When a proton tunnels within a base pair, it creates a tautomeric form of the base with different pairing properties. For example, cytosine after tunneling can sometimes pair with adenine instead of guanine. If this unusual pairing occurs during replication, it leads to a mutation in the new DNA strand. This quantum tunneling mechanism explains spontaneous mutations that classical models struggle to account for. While DNA repair enzymes correct many quantum-induced errors, some slip through, contributing to genetic variation. Studies suggest quantum entanglement exists between base pairs in a double helix. Entanglement links particles so their quantum states cannot be described independently. A single quantum state describes the system regardless of separation distance. In DNA, entanglement occurs between electron clouds of adjacent base pairs. The pi electrons of neighboring bases become quantum mechanically linked, creating extended quantum states spanning multiple base pairs. This entanglement allows quantum information to be shared across DNA instantaneously. This quantum property might explain how DNA maintains stability despite environmental threats. Entangled states could distribute potentially damaging forces across the molecule, preventing localized damage from disrupting the genetic code. Quantum coherence represents another fascinating aspect of DNA's quantum behavior. Coherence enables quantum systems to maintain consistent phase relationships between different states. The wave-like aspects remain synchronized, allowing constructive or destructive interference. Research indicates quantum coherence explains long-range interactions along the DNA strand. Classical models cannot easily explain how information transmits along DNA over distances greater than a few base pairs, as the structure typically prevents direct chemical interactions between distant regions. Quantum coherence allows quantum states to extend over longer DNA sections, enabling quantum telegraph effects. These coherent states might facilitate protein recognition of specific DNA sequences for gene regulation. Proteins must locate target sequences among billions of base pairs, an extraordinarily time-consuming task if relying solely on random diffusion. Quantum coherence could create distinctive quantum signatures for different sequences, allowing more efficient target location. Experimental evidence for DNA quantum effects continues to emerge. Advanced spectroscopic techniques have detected quantum coherence in laboratory DNA samples using ultra-fast laser pulses to excite molecules and measure resulting quantum coherent states. While maintaining these states is challenging in cellular environments, DNA's structure might protect quantum states from decoherence. The double helix creates a relatively isolated environment for base pairs, shielding them from external influences. Additionally, ordered water molecules bound to DNA might help maintain quantum coherence rather than destroy it. Theoretical models suggest structured water around biomolecules can support quantum effects. If these quantum properties are functional aspects of DNA, it suggests DNA operates as a quantum information processor. Classical information processing uses bits that are either 0 or 1, but quantum processing uses qubits existing in superpositions of both values simultaneously. This allows quantum systems to process multiple possibilities in parallel, potentially certain problems faster than classical computers. DNA's 4 nucleotide bases combined with their quantum properties could form a quaternary quantum code rather than a binary code. Each base pair position can represent multiple quantum states simultaneously, giving DNA exponentially greater information processing capacity than possible with classical biochemistry alone. Researchers have developed mathematical models of quantum teleportation protocols in DNA, showing how quantum information could transfer along the strand using entanglement. According to these models, RNA polymerase could act as a decoherent shield, preserving quantum states during transcription, allowing quantum information transfer from DNA to RNA during gene expression. If DNA functions as a quantum information processor, it transforms our understanding of genetics and molecular biology. The genetic code might contain layers of quantum information beyond the classical sequence of bases, potentially explaining observed complexity in living systems. These findings about DNA's quantum nature revive philosophical questions about life's origins and add new dimensions to the debate about evolution versus design. As research continues, we find ourselves at the intersection of quantum physics, information theory, and biology, opening new avenues for understanding life itself and developing technologies inspired by nature's quantum processor, the modern watchmaker argument. In 1802, the English theologian William Paley published a book called Natural Theology that would shape discussions about biology and design for centuries to come. In it, he presented what became known as the watchmaker argument. Paley asked readers to imagine walking across a field and finding a watch on the ground. Upon examining its intricate gears, springs, and precise movements, any reasonable person would conclude that this complex mechanism was designed by an intelligent creator, a watchmaker. Paley then extended this reasoning to living organisms, arguing that their complexity similarly implied the existence of a divine designer. This perspective dominated biological thinking until 1859, when Charles Darwin published On the Origin of Species, presenting a theory that fundamentally challenged Paley's reasoning. Darwin's theory of natural selection provided an alternative mechanism for the development of complex biological structures without requiring a conscious designer. Through the accumulation of small beneficial variations over immense periods of time, natural selection could gradually shape seemingly designed features. Darwin's explanation transformed biology by showing how undirected natural processes could produce the appearance of design. Complex structures like eyes could evolve gradually from simpler light-sensitive cells, with each small improvement providing a survival advantage. Over millions of generations, this process could produce structures of remarkable complexity and apparent purpose. Natural selection offered a powerful counter to the Watchmaker argument. Unlike watches, living organisms reproduce with variation and inherit traits from parents. With these crucial differences, biological systems could develop complexity through evolutionary processes rather than conscious design. For many scientists, this explanation eliminated the need for a designer in biology. The landscape of this historical debate has dramatically shifted with recent discoveries in quantum biology. What was once a discussion about mechanical structures visible to the eye has transformed into an exploration of life's quantum foundations, moving from gears and levers to qubits and quantum algorithms. Quantum biology reveals that life operates according to quantum mechanical effects at the molecular scale. These quantum properties include phenomena like electron tunneling and photosynthesis, quantum coherence and energy transfer, and potentially quantum information processing and DNA. These discoveries show biological complexity extending to the subatomic level. Proponents of intelligent design have formulated updated versions of the Watchmaker argument based on these findings. While Paley focused on mechanical structures visible to the naked eye, modern design arguments point to molecular machinery operating according to quantum principles that weren't even discovered until the 20th century. These updated design arguments emphasize information rather than mechanical complexity. DNA functions as a potential quantum information processor, storing and manipulating genetic information using quantum principles. Intelligent design advocates argue that such sophisticated information processing systems are unlikely to arise through unguided processes, similar to how complex computer code requires a programmer. The intelligent design movement has incorporated quantum biology by suggesting that quantum complexity in biological systems points to an intelligent cause. They argue that intricate quantum phenomena observed in biological processes, such as quantum coherence and photosynthesis, are too complex to have arisen by chance. This approach updates Paley's argument with modern scientific findings, focusing on information processing capabilities rather than mechanical structures. The scientific community has generally responded skeptically to these updated design arguments. Most biologists and physicists maintain that quantum effects in biology, while fascinating, don't necessarily point to design. They argue that natural selection could have discovered and optimized quantum effects through the same evolutionary processes that shaped classical biological structures. Scientists also point out that our intuitions about design may be misleading when applied to quantum systems. Human intuition evolved to understand macroscopic objects following classical physics, not quantum phenomena. What seems impossibly complex from our classical perspective might be a natural consequence of quantum interactions we're only beginning to understand. Modern design arguments typically avoid explicitly identifying the designer as a religious deity, allowing for various possibilities, including advanced extraterrestrial beings with sophisticated quantum engineering capabilities. This represents a shift from Paley's explicitly theological argument to one that attempts to function within scientific discourse, though most scientists remain unconvinced of its scientific validity. What's particularly fascinating is how quantum biology has transformed the design debate from mechanical analogies to questions about information processing and quantum programming. The Watchmaker's precision tools have been replaced by quantum code in our metaphorical understanding of life's complexity. This shift reflects the broader transformation in our understanding of life itself. We've moved from seeing organisms as intricate machines to viewing them as sophisticated information processing systems operating according to quantum principles. Regardless of where one stands on the question of design, quantum biology has added new dimensions to our understanding of life. The extraordinary complexity of biological quantum systems inspires wonder whether one attributes that complexity to natural processes, intelligent design, or some combination of factors. As we continue to uncover the quantum foundations of life, the philosophical questions raised by Paley over 2 centuries ago remain relevant, though transformed by our expanding scientific knowledge. The Watchmaker argument hasn't disappeared, it has evolved, much like the biological systems it seeks to explain. But to evaluate these competing explanations properly, we must consider how they measure up against established scientific criteria, particularly the requirement of falsifiability. Scientific falsifiability in quantum design. For a theory to be considered scientific, it must meet a fundamental requirement established by philosopher Karl Popper in the mid-20th century, falsifiability. This principle states that for an idea to qualify as scientific, there must be some way to prove it wrong through observation or experimentation. A hypothesis that cannot be disproven, no matter what evidence might be discovered, falls outside the realm of science. This criterion helps separate scientific theories from philosophical or religious beliefs, which may be meaningful but aren't testable through scientific methods. This requirement creates a particular challenge when examining theories about historical events like the origin of life. We cannot travel back in time to directly observe how life began. Instead, scientists must develop hypotheses that make predictions about what evidence we should find today if their theory about the past is correct. These predictions must be specific enough that finding contradictory evidence would force a rejection or major revision of the theory. The hypothesis that DNA was engineered by advanced beings faces significant hurdles when evaluated against this falsifiability standard. For such a hypothesis to qualify as scientific, it would need to make specific testable predictions about what we should observe in DNA or biological systems that couldn't be explained by natural evolutionary processes. These predictions would need to be unique to the engineering hypothesis, finding evidence that could be explained equally well by both evolution and engineering wouldn't help distinguish between the competing explanations. For the quantum design hypothesis to be considered scientific, it must meet several key criteria, make predictions about biological features unexpected under evolutionary theory. And should these predictions are specific enough to test through observation or experimentation, specify conditions under which the hypothesis would be falsified, identify what evidence, if found, would prove the hypothesis wrong. Currently, quantum design proponents have struggled to meet these criteria. Most arguments for engineered DNA focus on the complexity of quantum biological systems and the improbability of their natural emergence. However, pointing to complexity alone doesn't constitute a falsifiable prediction. Evolution through natural selection provides a mechanism for complexity to increase over time without intelligent guidance. To distinguish design from evolution, proponents would need to identify specific features that could only result from intelligent engineering. The lack of falsifiable predictions represents the most significant scientific limitation of the quantum design hypothesis. Many arguments for engineered DNA take the form of this system is too complex to have evolved naturally, which is an argument from incredulity rather than a testable scientific claim. Such arguments don't specify what evidence would disprove the design hypothesis. If a natural pathway to quantum biological systems were discovered, proponents could simply claim the designer worked through natural laws. There are potential approaches that could test aspects of the quantum design hypothesis. Researchers might search for specific signatures of engineering in biological systems, patterns that serve no functional purpose but identify the creator. If DNA contained non-functional sequences that encoded meaningful information unrelated to biological function, this might suggest deliberate engineering. Reconstructing evolutionary pathways offers another avenue. Demonstrating step-by-step natural pathways through which quantum biological systems could evolve would weaken the design argument. Conversely, consistent failure to reconstruct such pathways, despite extensive efforts, might strengthen the case for design, though it wouldn't prove it conclusively. Scientific attitudes toward quantum biology itself have evolved significantly over recent decades. Initially, many physicists and biologists were skeptical that quantum effects could play important roles in biological systems. The warm, wet environment of living cells seemed incompatible with delicate quantum phenomena, which typically require carefully controlled laboratory conditions. Experimental evidence has overcome this skepticism. The discovery of quantum coherence in photosynthetic systems represented a turning point in quantum biology's acceptance. Researchers have documented quantum tunneling in enzyme catalysis and quantum effects in DNA. These observations have established quantum biology as a legitimate scientific discipline with growing experimental support. This evolution in scientific attitudes illustrates how initially controversial ideas gain acceptance through rigorous experimental testing and falsifiable predictions. Quantum biologists predicted specific quantum effects in biological systems and developed experiments to test these predictions. When confirmed, the field gained credibility. Could the engineering hypothesis follow a similar path? Proponents would need to move beyond arguments based on complexity and probability. They would need to develop specific testable predictions about what we should observe if DNA were engineered by advanced beings. Predictions distinct from what we would expect under evolutionary processes. Even if DNA functions as a quantum information processor, this doesn't necessarily indicate engineering. Natural selection could potentially discover and optimize quantum advantages through evolutionary processes. Just as eyes evolved to capture light, molecular structures may have evolved to capture quantum advantages. The quantum biology aspects of the hypothesis are increasingly supported by experimental evidence. Quantum coherence in photosynthesis, tunneling in enzymes, and potential quantum information processing in DNA are becoming established scientific facts. However, the design interpretation remains philosophical rather than scientific. Without falsifiable predictions distinguishing intelligent engineering from evolutionary processes, the design hypothesis cannot be empirically tested. This doesn't mean the hypothesis is wrong. It means it hasn't met the criteria for scientific theories. This distinction between quantum biology scientific status and quantum design's philosophical status highlights science's boundaries. Science addresses questions answerable through empirical testing. Some questions, including many about ultimate origins, may lie beyond these boundaries. When we reach these limits, we confront profound questions about our existence. How do we understand phenomena that science cannot fully explain? What does it mean if life's deepest mechanisms remain partially beyond our scientific grasp? If quantum capabilities weren't engineered by advanced beings, how did they arise? The primary scientific alternative suggests these remarkable quantum properties emerged through evolutionary processes operating over billions of years. Evolutionary explanations for quantum life. Evolution operates as a powerful natural algorithm, constantly testing variations and selecting those that provide survival advantages. We typically think of evolution selecting for traits like stronger limbs or sharper eyesight. But what if this same process has been selecting for quantum mechanical advantages throughout life's history? Natural selection operates at every level of biological organization, from entire organisms down to molecular interactions. The discovery of quantum effects in biological systems suggests the same evolutionary processes that shaped wings, eyes, and immune systems also optimized quantum properties in living organisms. For this evolutionary process to work, quantum effects must provide real survival and reproductive advantages. The evidence indicates they do. Consider photosynthesis, where plants and certain bacteria that capture energy more efficiently can produce more energy, grow faster, and outcompete their neighbors. Research now shows quantum coherence enables more efficient energy capture, giving organisms with better quantum coherence a significant advantage. Hashtag from small advantages to quantum optimization in the harsh competitive environment of early Earth. Even small efficiency improvements provided meaningful advantages. Organisms extracting slightly more energy from sunlight or performing enzymatic reactions marginally faster produced more offspring, gradually spreading these quantum enhanced traits through populations. This process resembles how software evolves through updates. Each version may contain small improvements that users barely notice, but over many iterations, the cumulative enhancements transform the entire system. Similarly, Quantum properties in biological systems evolved through countless generations of incremental improvements, each providing a small but significant advantage. The Fina-Matthews-Olsen FMO protein complex in green sulfur bacteria offers a compelling example of this evolutionary refinement. This complex exhibits quantum coherence that enhances energy transfer efficiency. Green sulfur bacteria often inhabit low-light environments where capturing every available photon is crucial for survival. In these conditions, the quantum advantage provided by coherence would be strongly selected for, as bacteria with more efficient energy capture would reproduce more successfully. Over countless generations, bacteria with mutations enhancing quantum coherence gradually outcompeted their neighbors, leading to the optimization of these quantum properties. The process required no understanding of quantum mechanics, just the blind algorithm of natural selection testing variations and preserving beneficial ones. Simulating quantum evolution. Computer simulations have helped scientists understand this evolutionary process. By creating virtual organisms with varying quantum properties and subjecting them to simulated environmental pressures, researchers observe how quantum advantages spread through populations over time. These simulations consistently show that quantum-enhanced traits can evolve through standard evolutionary mechanisms. One fascinating aspect of these simulations involves the evolution of quantum decoherence protection. Quantum coherence is typically fragile, easily disrupted by environmental noise. Yet biological systems maintain coherence in warm, wet environments where it should quickly disappear. Simulations suggest natural selection favors structures that protect quantum states from environmental disruption, explaining the remarkable robustness of biological quantum effects. #Time, evolution's most powerful resource. The timescales available for evolutionary optimization are vast. Life has existed on Earth for approximately 3.8 billion years, providing ample opportunity for the gradual refinement of quantum biological mechanisms. Even extremely rare beneficial mutations would have time to arise and spread through populations over such extended periods. What might seem improbable over short time frames becomes almost inevitable when selection operates over billions of years. These extended time frames allow for the accumulation of complex adaptations through intermediate steps. Quantum effects in biology developed gradually, with each intermediate stage providing some benefit. This step-by-step process explains how complex systems emerge without requiring simultaneous appearance of all components. Catch has selection for function, not quantum nature. Selective pressures favor quantum-enhanced biological systems for their functional benefits. In addition to efficient energy capture in photosynthesis, quantum tunneling in enzymes allows for faster reaction rates. Similarly, quantum effects in DNA might enhance information, storage, or repair mechanisms, improving genetic fidelity. These quantum advantages are selected for because they improve survival and reproduction through their effects on biological function, not because they're quantum. Natural selection has no awareness of quantum mechanics. Only the impact on fitness matters. Quantum biology within evolutionary theory. Quantum biology fits comfortably within modern evolutionary synthesis. Rather than challenging evolutionary theory, it extends our understanding of evolution's mechanisms by adding another layer to evolutionary processes without contradicting fundamental principles. The discovery of quantum effects in biology reveals that evolution has been optimizing biological systems at both macroscopic and quantum levels. This optimization requires no conscious design, just the patient operation of natural selection over immense time periods. The evolutionary explanation for quantum life doesn't diminish its wonder. Realizing that sophisticated quantum systems emerge through natural processes deepens our appreciation for life's remarkable complexity. The quantum properties that make biological systems so effective were discovered by the patient exploration of natural selection, testing countless variations over billions of years. This evolutionary understanding of quantum biology opened exciting possibilities for technology. By studying how biology maintains quantum coherence at room temperature or utilizes quantum tunneling in enzymes, we might develop new approaches to quantum computing and energy capture. Nature's quantum solutions, refined over billions of years of evolution,
Biomimicry,
may
learning
hold
from
keys
nature's
to our own
quantum
technological
technology.
advancement.
While engineers have spent decades struggling to build functional quantum computers in specialized laboratories, nature seems to have mastered quantum technology billions of years ago. Living cells might hold the solutions to our most challenging quantum engineering problems. This realization has sparked an entirely new approach to quantum technology development, looking to biology for inspiration. Biomimicry applies nature's designs to solve human problems. With our understanding of quantum biology expanding, we recognize that living systems have already created the quantum blueprints we've been struggling to develop. Plants, bacteria, and human DNA perform quantum operations that function in conditions our engineers can only dream of: room temperature, wet environments, without extreme cooling or isolation. Photosynthesis stands as nature's quantum masterpiece. Plants and certain bacteria convert sunlight into chemical energy with efficiency that outperforms our best solar technologies through quantum coherence, allowing captured photon energy to explore multiple pathways simultaneously before choosing the optimal route. Scientists are now reverse engineering this natural technology for artificial photosynthesis systems that could convert sunlight directly into fuels like hydrogen. The Pheno-Matthews-Olsen protein complex in green sulfur bacteria serves as their primary blueprint, demonstrating nearly perfect energy transfer efficiency through quantum coherence at room temperature, a stark contrast to our quantum computers requiring temperatures near absolute zero. Minus 273.15 degrees Celsius. The implications extend beyond energy harvesting. DNA functions as a sophisticated information processor with electron cloud arrangements, creating quantum states that enable advanced information processing. Quantum tunneling occurs during DNA mutation and repair, while quantum entanglement may exist between base pairs and the double helix. These biological quantum computers are inspiring new computing approaches. While traditional quantum computers use superconducting circuits or trapped ions as qubits, DNA-inspired systems could potentially use organic molecules that self-assemble and operate without complex fabrication processes. Early DNA computers already perform calculations by manipulating DNA strands, though they don't yet fully harness quantum effects. The remarkable resilience of biological quantum systems offers perhaps the most valuable lessons. Rather than fighting environmental noise, biological systems appear to use it advantageously, with protein vibrations actually enhancing quantum effects instead of destroying them. Researchers recreating these protective environments could develop quantum devices operating without extreme cooling or isolation, bringing quantum technology from specialized labs into everyday applications. Imagine a future where these principles transform medical diagnosis. A handheld quantum sensor inspired by plant proteins could analyze your breath, detecting disease markers with molecular precision. Your physician might use quantum imaging technology derived from biological sensing systems to visualize cellular processes with unprecedented clarity, identifying problems before symptoms appear. In this future, your home might be powered by solar panels that incorporate organic molecules arranged to maximize quantum energy transfer, doubling or tripling current efficiency rates. The quantum computer on your desk might process information using principles derived from DNA, operating silently at room temperature without specialized cooling systems. Medical applications represent another frontier. Quantum sensors based on biological principles could detect disease biomarkers with extraordinary sensitivity. Some researchers believe smell involves quantum effects, with molecules recognized by quantum vibrational signatures. This understanding could lead to quantum noses capable of detecting diseases through breath analysis. Energy research stands to benefit enormously from this biological inspiration. Beyond artificial photosynthesis, researchers are examining how quantum effects and photosynthetic bacteria could inform new materials for solar technology that dramatically outperform silicon-based systems. The computing implications remain perhaps most significant. Rather than requiring massive infrastructure, vacuum chambers, cooling systems, and isolation from interference, bio-inspired quantum computers might eventually sit on desktops, not entire laboratories. Transferring these principles to practical technology isn't straightforward. Biological systems evolved over billions of years through countless generations. Our understanding of how these quantum effects work remains incomplete, and adapting them requires more than mere copying. We must understand the fundamental mechanisms well enough to implement them in new contexts. Nevertheless, quantum biology research has already yielded insights changing our technology development approach. The discovery that quantum coherence can persist in warm, wet environments challenges previous assumptions about what's physically possible. By studying natural quantum systems, we may find breakthroughs for practical quantum technologies under ambient conditions. The future of quantum technology may resemble the elegant, self-assembling molecular machinery and living cells more than today's sterile laboratory setups. Nature has spent billions of years refining its quantum technology, and we're finally beginning to decode its instruction manual. This convergence of quantum physics and biology raises profound questions about life itself. If quantum effects are fundamental to biological functions, what does this reveal about life's origins and evolution? As exploration continues, we're discovering not only new technologies, but new perspectives on living systems in a quantum universe. The future of quantum biology research. We're standing at the edge of a new scientific frontier. Quantum biology research is expanding rapidly, reaching beyond the well-established examples of photosynthesis and DNA to explore how quantum effects might influence many other biological processes. As one research scientist recently noted, quantum biology is attracting significant investment from technology giants, pharmaceutical companies, and startups, with transformative changes expected across healthcare, semiconductors, artificial intelligence, and bioinformatics within the next 2 decades. The tools we need to probe deeper into biological quantum effects are becoming increasingly sophisticated. New quantum sensors can detect biological signals with unprecedented sensitivity. Advanced spectroscopic techniques can now track quantum processes occurring on timescales of femtoseconds, billionths of a millionth of a second. Quantum imaging technologies are pushing the boundaries of what we can visualize at the molecular level. These technological advances allow scientists to test hypotheses about quantum effects in biological systems that were previously impossible to investigate. One of the most provocative areas of quantum biology research involves consciousness itself. The orchestrated objective reduction, orchestral theory, proposed by physicist Roger Penrose and anesthesiologist Stuart Hameroff, suggests that consciousness emerges from quantum processes in the brain's microtubules, protein structures within neurons that might sustain quantum coherent states long enough to influence neural activity. Could these microtubules function as natural quantum computers, with their collapse into definite states generating conscious experience? While this theory remains highly speculative, it has inspired experiments to test whether quantum coherence can exist in the warm, wet environment of the brain. The sense of smell represents another frontier in quantum biology. The conventional understanding suggests molecules are identified by their shape, but this model struggles to explain how we distinguish between molecules with identical shapes but different vibrational properties. The vibrational theory of olfaction proposes that our sense of smell might detect quantum vibrational signatures of molecules through electron tunneling, generating signals our brain interprets as specific smells. Recent experiments with fruit flies provide tentative support for this idea, potentially revealing a quantum mechanism operating in one of our primary senses. Enzymatic reactions throughout your body might also harness quantum effects more extensively than previously recognized. Research has established that quantum tunneling occurs in some enzymes, allowing particles to pass through energy barriers rather than over them, a quantum shortcut that dramatically enhances enzyme efficiency. The enzyme lactate dehydrogenase, crucial for cellular respiration, uses quantum tunneling to catalyze key metabolic reactions. Might enzymes create protected environments where quantum coherence persists long enough to coordinate complex reactions? Understanding these mechanisms could lead to designing pharmaceutical compounds that target specific quantum processes in enzymes associated with disease. Medical applications represent a promising direction for quantum biology research. Future quantum sensors might identify disease biomarkers at concentrations too low for conventional tests, enabling earlier diagnosis of conditions like cancer. Quantum imaging techniques could enhance medical visualization with higher resolution images and lower radiation exposure. Other quantum diagnostic tools could analyze biological samples using principles observed in quantum biological systems, offering more comprehensive health assessments. Understanding diseases through a quantum biological lens could fundamentally change treatment approaches. Some researchers speculate that certain conditions might involve disruptions to quantum processes in cells. If DNA repair mechanisms rely partly on quantum tunneling, defects in these quantum pathways could contribute to cancer development. Similarly, if protein folding involves quantum effects, misfolded proteins in conditions like Alzheimer's might represent quantum processing errors rather than classical chemical mistakes. The potential for quantum biology to transform medicine brings important ethical considerations. Advanced quantum diagnostic tools might initially be available only to wealthy individuals or nations, potentially widening existing health inequalities. Will we navigate this quantum medical revolution more equitably than previous technological advances in healthcare? What makes quantum biology particularly exciting is its interdisciplinary nature, requiring collaboration among physicists, biologists, chemists, computer scientists, and medical researchers. This collaborative approach has already produced remarkable insights, from quantum coherence and photosynthesis to new models for investigating potential quantum effects in brain function. Looking forward, quantum biology represents a scientific paradigm shift that may fundamentally change our understanding of what life is. If quantum effects prove central to processes like photosynthesis, enzyme function, DNA stability, and potentially consciousness, we may need to reconceptualize living systems as quantum mechanical phenomena rather than purely classical chemical machines. Much as early astronomers peering into the cosmos forever changed humanity's place in the universe, might quantum biologists reshape our very understanding of life itself? The philosophical implications are profound. If quantum processes underlie consciousness, what does this tell us about the nature of mind? If quantum effects operate throughout living systems, how does this change our understanding of the boundary between physics and biology? These questions have motivated a new generation of scientists to explore the quantum foundations of life. As we stand at this frontier, we see the potential for quantum biology to transform medicine, inspire new technologies, and deepen our understanding of life itself. The quantum revolution that transformed physics in the early 20th century may now be extending to transform biology in the 21st. The quantum nature of DNA changes everything we know about life, whether through random mutation and selection over billions of years or deliberate engineering by advanced beings. This discovery represents one of science's greatest unexplored territories. These quantum processes within our cells compel us to reconsider life at its core. Photosynthesis, enzyme function, and possibly consciousness operate according to principles that merge physics and biology. 75 years after Schrödinger asked, "What is life?" his intuition proves correct. The quantum classical boundary runs directly through our cells, unlocking a hidden code that might forever transform our understanding of existence. What other biological mysteries might quantum mechanics solve?